Getting close to the end now. This topic is cardiovascular physiology. Couple things. First, it is a bunch of little logical relationships, and you really have to understand the first ones that I present before you go on with the next ones. So the reason I'm telling you that is because you need to digest this in small chunks. You need to make sure that you are consistent with yourself about assessing whether you actually understand it before you move on. And then you need to ask lots of critical questions, either in the discussion of one another or just of yourself. So let's start with just acknowledging that with this particular set of material, there's a lot of before you begin, because you really have to make sure that you are familiar with cardiovascular anatomy before you can go on with cardiovascular physiology. And I'm not saying you have to remember every single superficial and deep vein of the upper limb, but you have to remember the cardiovascular anatomy of the heart, the systemic circulation and the pulmonary circulation, where blood is oxygenated, where it's deoxygenated, what the valves do. So to that end, there is a relatively long before you begin section here, almost 2 pages long, with figure references. Also in interactive physiology, there's a really good review of the anatomy of the heart. It's almost 6 minutes long. It's a good review. Make sure that you feel confident on that stuff before you move on to the phys, because the phys depends on the anatomy. I mean, it always does, but with cardiovascular system, it really does. Okay, so what I'm going to do is I'm going to start at Roman numeral 2, assuming that you have reviewed the anatomy. We are going to start with Roman numeral 2, which is sort of the physiological plan or design of the cardiovascular system. I'm not meaning that it was designed. I'm meaning that what is it set up to do, so that you can understand the big picture. So it's all a story related to pressure, flow, and resistance. Ultimately, the overriding function of the cardiovascular system that all of the other things serve is that the cardiovascular system is supposed to ensure adequate blood flow to capillary beds, no matter what the cost. So for instance, if you had a capillary bed that had really low flow, you would bump up the pressure even if it damaged the pump or the tubes, because the overriding functionality, the one that trumps all of the others, is the ultimate function of the cardiovascular system is to ensure adequate blood flow through capillaries. And you need to remember that, because if, for instance, the blood pressure gets so high that it's damaging the tubes or the pumps, but it's still not delivering adequate blood flow to capillary beds, you will continue to increase the blood pressure. Because if you think about it, if you don't have adequate blood flow to capillary beds, you're going to die anyway, because of course the blood flow is where the nutrients and the oxygen are being delivered. No nutrients and oxygen, no cellular respiration, no ATP, no ATP, no homeostasis. Okay, so remember that is the function that trumps all others. Okay, so things that you know, putting into context here. All the gas and nutrient exchange happens at the capillary beds. Why? Because capillaries are branched enough with enough surface area and thin enough to allow for gas, nutrient, and waste exchange. So every single cell in the body is within a few centimeters of at least one of the capillaries. And of course, if we're talking about systemic capillaries, what they're typically doing is they are bringing to the capillary bed nutrients and oxygen, and they are taking away from the capillary bed wastes and carbon dioxide as a result of course, cellular respiration primarily. So a couple of concept check questions. How do oxygen and nutrients typically get from the capillaries to the interstitial fluid to the tissue? They are going to move out primarily by diffusion. Sometimes there's transport mechanisms as well. But diffusion from high concentration to low concentration, especially if you think about gases will move from the systemic arteries, where they're relatively high concentration of oxygen, into the capillaries, where it's going to be relatively low because they've been using it, right? Move in. Carbon dioxide is going to move from the cells, where it's being generated via cellular respiration, into the capillary bed, where it's relatively low because we didn't bring appreciable amounts of carbon dioxide into the tissue. A little bit of solutes are actually pushed out by filtration as well, simply because the blood that you bring through the arteries is going to be high pressure. Okay, waste products are generally going to move from the interstitial fluid back into the blood by diffusion, mediated transport if necessary. And because filtration is going on, and filtration is moving things from high pressure to low pressure, and capillaries are always bringing in relatively high pressure blood, you are going to lose water by filtration. A lot of the water that's lost by filtration will get picked back up by osmosis because the blood plasma has a relatively high concentration of solutes compared to the interstitial fluid. And some of the water will also get picked back up by the lymphatic system. Okay, so now let's just talk a little bit about the overview of blood flow, which is a lot of what this chapter is going to be about, or this set of notes is going to be about. So why does blood move? Big picture understanding. I mean, it's really very easy to understand if you just think about plumbing. Blood moves, like all fluids move, from high pressure to low pressure. And so if we're looking at systemic circulation, that will be highest pressure in the left ventricle. From high pressure to low pressure, blood flows. Blood moves due to bulk flow produced by pressure. The pressure is created by the pumping of the heart, of course. And the pattern that you are going to see almost always is from ventricles to arteries to arterioles to capillaries to venules to veins, and then back to the heart. And the pressure differential goes from about 85 millimeters of mercury in the biggest arteries near the heart, think aorta, to the vena cava, where it's about 0, and the pressure falls, and therefore the blood flows unidirectionally. Okay, so as pressure increases, what do you think tends to happen? These are some tendencies. As pressure increases, what do you think tends to happen in the system? As pressure increases, flow tends to do what? Flow tends to increase until pressure gets above normal physiological levels, and then increased pressure will decrease flow, but we'll talk about that later. The general trend with an asterisk is that as pressure increases, flow tends to increase. And sort of a way to think about that is to think about the pressure in your pipes. If you increase the pressure, turning up the water pressure, the flow tends to increase. Flow, though, is also opposed by what we call resistance. And resistance is basically the opposition to flow that usually occurs inside the tube. Like if the tube was really narrow or if the tube was really gunky, flow is opposed by what we call peripheral resistance. So if resistance opposes flow, what do you think the relationship is? As resistance increases, flow tends to do what? Flow tends to decrease. Resistance occurs primarily in the vessels, and so we're about to get into that in a little bit more detail. Okay, so the relationship that we are going to see is that flow equals the change in pressure, or the difference in pressure between 2 points over resistance. So let's look here and do a little whiteboard. F, which is flow, delta P equals, difference in pressure between points. And I'll tell you what I'm talking about in just a second. And R equals resistance. So let's make a theoretical blood vessel for a second. And say, okay, here is a blood vessel, and here is point 1, and here is 0.2, each of which has a pressure associated with them. You probably know, but I'll just go ahead and tell you that pressure is measured in millimeters of mercury. So if I say the pressure at 0.1 is 100 millimeters of mercury, and the pressure is 0.2, at 0.2 is 80 millimeters of mercury, then delta P equals, delta P equals 20. Okay, so really what this means is that it's not the absolute pressure, it's the difference in pressure. Because if the pressure at 0.1 and the pressure at 0.2 were identical, you'd have no flow at all. But I'll elaborate on that in just a second. Okay, so flow of blood is determined by two factors, by pressure and resistance. So I want to talk about pressure a bit. Let's look at this figure. Okay, so blood, like all fluids, is going to flow from high pressure to low pressure. If you turn on your tap, the pressure is lower in your sink than it is in your faucet. Okay, and so the fluid flows from high pressure to low pressure. Pressure is, as I mentioned, measured in millimeters of mercury. And then pressure is generated by cardiac muscle contraction. So the cardiac muscle contraction, specifically ventricular contraction, is responsible for generating the pressure in the system of tubes. And that's called cardiac output. We'll cover it in more detail later. So pressure is generated by what the pump is doing, but it's dramatically impacted by the volume of fluid in the tubes. So a little bit of fluid in the tubes, low pressure, more fluid in the tubes, high pressure. So it's dramatically affected by blood volume. Okay. And then also it can be impacted by the size of the tubes. So the lumen diameter, if it's smaller, it's going to be higher pressure. If it's bigger, it's going to be lower pressure. So this is the general relationship between pressure. So let me show you what I mean about the whole delta P business in just a second. So before I kind of told you that higher pressure favored flow, but what's really true is that it's actually the difference in pressure between 2 points. So it's the difference in pressure between 2 points that determines both the direction of flow and the velocity of flow. So stick with me. There's three situations that you're seeing here in this figure. Okay. They are holding the resistance constant here because they're good scientists, even in a diagram, and you only manipulate one variable at a time. So the resistance is set at arbitrarily 2 here. So, but we're going to have three different situations. In three different situations, we are running into here, let's call all of the left ones 0.1 and all of the right ones 0.2. Okay, so in situation A, the pressure at 0.1 is 100 millimeters of mercury, and the pressure at 0.2 is 60 millimeters of mercury. So the delta P, the difference in pressure between 2 points, is 40 millimeters of mercury. So that will let us know which direction the fluid is going to flow. It flows from high pressure to low pressure, right? Okay. And also it will, if you factored in resistance, it would tell you the velocity, which would end up being flow equals delta P over R. And if R was arbitrarily set at 2, this would give you 20 mils per minute. So just hold on to that for just a second. But in this situation, importantly, it doesn't matter what the delta P is. And they look like they're both maybe 60 here. Doesn't say. 60 millimeters of mercury, 60 millimeters of mercury. The delta P is 0. And the flow is 0. So it's delta P over R. You get no flow whatsoever, even if you increase the pressure on both sides, because it's not the pressure that matters, it's the difference in pressure between 2 points. So no pressure whatsoever. And then let's look at figure C, because in figure C, we're actually getting the same flow that we did in figure A, but we're getting it at much higher pressures because, again, it's not the absolute pressure that matters. It's the difference in pressure between 2 points. So this one, 0.1, has 200 millimeters of mercury, and 0.2 has 160 millimeters of mercury. Still, the delta P is 40 millimeters of mercury, and so the flow is the same. So this is kind of analogous to what's the blood flow in a person with normal blood pressure. And how could it be the same as the blood flow in a person with hypertension? Well, in a person with hypertension, the tubes are so high because of maybe vasoconstriction and arteriosclerosis. Maybe it's all gunked up, and so the tubes are really high pressure. So what has to happen is you have to generate higher pressure with the heart to get over it. So again, the point of this figure right here is it's not the absolute pressure in the system that matters. It's the difference in pressure between 2 points that tells you both the directionality of flow and helps you figure out the speed of flow. I mean, you have to factor in resistance as well, but it'll help you figure out the speed of flow. Okay, so the truth, to clarify a little bit more about what we said in pressure and flow before, is that flow is directly proportional to the difference in pressure between 2 points, or not as pressure increases, flow increases, but as pressure difference increases, flow increases. Okay? And of course, you know and you understand this intuitively, that the magnitude of the pressure in the system is going to vary. We saw this throughout systemic and also pulmonary circulation. So it's going to be highest in the left ventricle, lowest in the right atrium. It's going to fall as you move through systemic circulation. It actually does as well through pulmonary. It's just the pressure in the whole pulmonary system is lower. So here is looking at the average pressure throughout the system. And since the pressure is highest in the arteries, blood will flow from the arteries to the arterioles, to the capillaries, to the venules, to the veins, to the superior and inferior vena cava. That's in the systemic circuit. In the pulmonary circuit, it does the same thing. Way lower pressure because, of course, the right ventricle is much less muscular and generates less pressure than the left ventricle. But the trend is the same in both the systemic and the pulmonary circuit. So the average pressure falls throughout the two circuits as blood flows from arteries to arterioles, capillaries, venules, veins. The average pressures are highest in the arteries, but they're also most variable in the arteries. So let's look, instead of at average pressure, let's take a whole bunch of data points and look at just the systemic circuit and look at how the pressure actually is, not average pressure. Okay, so the pressure is highest in the arteries for sure. The average pressure is highest, but it's also very variable in the arteries. And so this accounts for systolic and diastolic pressures. So you guys have done this in lab, but let me reiterate because it may have been a minute since you've done it. This is the pressure in a systemic artery. I don't know, let's say the brachial artery. Immediately before systole, which is diastole. And this is the pressure in the brachial artery, maybe immediately after ventricular systole. So it's super variable. It drops to, let's say, 80. That's the diastolic pressure, and then goes up to 120, 80, 120, 80, 120, 80, 120. These are the pulse pressures that you are measuring in an artery. Notice that it is still variable in arterioles, right? Still a little bit variable at the arterial end of a systemic capillary bed. And then as you go through the capillary bed, you are going to lose that variability, and it's just basically going to drop and be relatively steady as you get into a venule and into a vein. So again, the point of this figure is really that the average pressures are highest in the arteries, but the pressure is also most variable in the arteries. You call the average arterial pressure the mean arterial pressure, and there's a way to calculate that. And then the pressure is lowest in the veins. Again, one more time, the pressure is really impacted primarily by three things: cardiac output, which is what the heart is doing, blood volume, which is how much fluid is in the tubes, and then what we call vessel diameter, which is just how big the tubes are. Okay, I'm going to stop right there, and then I'll do resistance in the next one.